Welcome everybody to today's webinar on the control of filamentous foaming. I'm Dan McKeaton. I'm the Director of Wastewater Education here at Aquafix. I'm also a chemist and a researcher here. I work with Aquafix Laboratories. So Aquafix Laboratory is a lab that provides customer service and consultation. We do microanalysis and reports for customers, that kind of thing. We also handle all of our research and development for products and just in general to understand wastewater problems. If you have any questions that you'd like to talk to the lab about addressing related to your system, definitely reach out to us. We're happy to answer any questions that come up, and we're always happy to look at your samples when they come in. All right, so most people have replied to the poll at this point. We're seeing 51% Nocardia, 22% Microthrix. You know, that's pretty typical. Interestingly, I read once in Europe, they tend to deal more with Microthrix, and in the United States, we deal more with Nocardia. I'm not sure why that is, but kind of an interesting point here. The materials provided. These are a couple sheets you'll have in the handouts tab in your chat. You can access these at any time during the presentation. If something comes up that you think you might want some literature on, reach out to us. If we have any literature, we'll send it your way, or at least we'll be willing to discuss whatever your question is. So today we're going to be covering a number of things. First, we're going to be doing a little bit of work with field and microscopic identification of common foaming filaments. So we're going to talk about how on-site you can identify a filamentous foam versus surfactant foam and how reliable that sort of thing is. We'll also briefly touch on staining and microscopic analysis to determine the ID of organisms. Then we're going to go through the reasons foaming filaments show up, pros and cons of various chemical control strategies, and then we'll talk about how our Aquafix products and different operational changes may help to improve system health and help your bacteria to outcompete filaments. Starting out, you'll see some foam in your site, and then you have to be, wondering, all right, is this a filamentous foam? Is it a surfactant foam? Depending on how you handle each of them, it may result in dramatically better results, or you can actually make the problem worse. So you have to be a little careful. But on the left here, you have this white fluffy foam. That is pretty typical of a surfactant or a young sludge age foam. Both of these kind of operate similarly. The reason it is white in color is because there tends to be very low solids in the foam. It tends to be mostly just the surfactant mixed with air and a little bit of water in there. So these white fluffy foams tend to show up quickly and go away quickly. Whereas on the right here, you could see a foam like that just from having a lot of grease in your system, but generally that is sort of stereotypically a filamentous foam. So surfactant foams, you'll look for fluffy light color, 1 to 3 feet thick, and it tends to show up within hours. Generally it doesn't go on weeks, but sometimes it could. But generally this type of foaming just shows up, then it'll go away in a couple days. You won't necessarily know where the surfactant came from. Whereas the biggest thing to consider here is filamentous foaming tends to stick around for months without any treatment, and it can get very, very bad. It's a lot harder to collapse filamentous foam. It's a lot more dense. Generally, it's darker in color. One thing I should touch on, since I'm talking about looking at the foam and seeing if it is a filamentous foam or a surfactant foam, generally speaking, these rules hold true probably about 90% of the time. But there have been a couple flukes we've run into where we've had a white fluffy foam that is actually the result of filaments. And we've also had a couple cases where we've had sort of a dense foam that was actually the result of surfactants. And these are generally in higher solid system. But just remember, when you're looking at a foam and you're not looking at it under the microscope, you can be relatively sure you're identifying it somewhat appropriately, but you can't be 100% sure. So keep that in mind. All right, filamentous bacteria. We have a nice picture here of a lot of filaments going on. These are a nice tangly filament on the right. This is another thing I should note while we're here. Generally speaking, filamentous organisms can look very similarly if you're looking at them in lower magnifications. It can be very helpful to look at them in a little bit higher magnification so you can get a sense for things like cell shape. For example, Microthrix parvicella, which many of you will deal with, tends to be sort of a tangled spaghetti-like filament. So we had a customer or a client identify this filament in the picture as Microthrix parvicella. It is actually not. It's too big. It has very distinctive cells, whereas Microthrix, you can't really see cells in higher magnifications. And the way they were treating their system actually ended up encouraging the growth of this filament rather than discouraging it. So you got to be a little careful. Another thing on filaments, a lot of the time they are good. Filaments are not necessarily a bad thing at all, but if you have them at excessive levels in your system, they can cause major issues. So just keep them in mind. Generally speaking, they'll cause foaming and bulking. Our foaming filaments are Microthrix and Nocardia forms most of the time. There's a couple others that can cause foaming in a number of circumstances. Basically, these foaming filaments work by trapping air and mixed liquor and rising to the surface, and then you get your fluffy foam. So one thing you can do to improve your certainty when you're doing a microscopic analysis to determine what type of filament you're dealing with is to do a stain. Now, these aren't incredibly difficult to do. If you don't feel comfortable with doing them, that's totally fine. There's a little bit of finickiness going on in the decolorizing step of the gram stain, so you can accidentally make your filaments appear gram negative or positive. So to go over the terms in this gram stain, gram-positive filaments tend to stain a purpley color like the Nocardia forms in this image. This purpley color means it is a gram-positive organism. That is caused by this first stain on our list, which is Graham's crystal violet, which stains everything in the mixed liquor sample this purple color. The next phase in the gram stain, you apply the iodine from the gram stain, and it will bind the crystal violet to gram-positive organisms. And then you follow that up with a decolorizing step, which washes off the crystal violet stain from all of the things that are not gram-positive, therefore separating gram-positive and gram-negative filaments. And then you apply a counter stain, which is Graham's safranin stain, which makes all of the non-gram-positive things appear kind of a reddish color. And that gives you a little better contrast, and it helps to cover up any residual gram stain that may be present on the filaments. This takes about 5 minutes. Generally, you add the dyes for about 1 minute each, and you decolorize for just a few seconds. You're going to kind of wash for the color running off the cell. So if you're interested in doing a little bit more with microanalysis, this is a very nice stain because foaming filaments tend to stain very strongly gram-positive most of the time. Sometimes Nocardia is a bit blotchier. So our first filament, we're going to talk about Nocardia. Now, somebody asked earlier in the presentation to touch on washout rates for filaments. That's an interesting question. So generally speaking, Nocardia forms and Microthrix, our foaming filaments, have a low density fatty cell wall, which causes them to float to the surface of a basin. But some of the filaments are going to remain suspended in the free liquid in your mixed liquor, and some of them will be trapped inside of flock. The filaments that are trapped inside of flock always have potential to stay in your system just like the flock do, whereas the ones that are free in the bulk liquid may be washing out of your system at a fairly steady rate relative to your hydraulic retention time. But they tend to be the result of filaments growing somewhat rapidly. So if you have active filament growth, you don't really see a washout per se. Your foam will kind of just sit there and be trapped in by any sort of barrier that covers the surface of your liquid. One thing we're going to touch on a couple of times, if you actually use something like a defoamer or spray water to collapse the foam, you'll actually be pushing the filaments from the foam back into your mixed liquor, which can cause them to repopulate it a little bit. So this one is pretty easy. Nocardia forms, Nocardia-like organisms. There was a bit of a terminology change we noticed in the last few months on this filament. So if I say Nocardia forms, that used to be what we consider to be most correct. Though for this presentation, I'll normally use the more casual Nocardia term instead of saying Nocardia-like organisms each time, because that gets a little wordy. So this is the only true branched wastewater bacteria that you'll see. One thing that is a bit confusing about Nocardia is there is a wide variety of different organisms that share the similar appearance. That's why they call them Nocardia-like organisms. They might be forms of Gordonia, Actinomyces. There's a wide variety of filaments that fit into this category, and they may have somewhat different behaviors, which you should keep in mind when you're treating something. For example, one Nocardia form might be a bigger fan of high levels of fat soils and grease, whereas one may be able to thrive with lower levels of fat soils and grease. Generally speaking, on the field, people say this is a dense, stable foam with a tan to brown color that can get up to several feet thick. I will say Nocardia tends to have much thicker foam than Microthrix, but in terms of the coloration and density, you can't necessarily tell the foams apart very easily. So here's kind of an early Nocardia outbreak. You can see a little bit of accumulation in the areas away from the aerators. This is not too bad, but if you were to see this in your system and you were to perform a microanalysis and you saw moderate levels of Nocardia forms in your mixed liquor, I'd say you should be concerned and maybe lower your sludge age a bit at this point before it turns into a real foaming emergency. We'll talk about why lowering sludge age works a little bit later. But it's good to catch this sort of thing as early as possible. All right, so here's a more advanced Nocardia foam. You'll notice there's kind of a thick scum covering the entire surface of the basin here. This can have another impact, which is it can prevent air diffusion into the surface of the liquid. So if you're preventing air diffusion, you can actually end up with low DO conditions in your basin. Now notice they are spraying water at the top of the screen. You can see they're spraying water on top of their foam. This does not work real well to collapse filamentous foam. Filamentous foam tends to be very difficult to collapse, and sometimes it'll push a little bit into the mixed liquor when you do it, like I said, but yeah, just keep that in mind. So our next foaming filament is Microthrix parvicella. We'll call it Microthrix for short. It is very gram-positive. You can see it's a very nice, deep purpley color here. If you're looking at it for an identification, it's considered to be spaghetti-like tangles. Now this filament, you need to be a little bit more careful if you see it under the microscope, because I showed the example earlier. Microthrix tends to be a little bit more difficult to identify definitively without staining. In fact, it actually helps to do a second stain, which I didn't talk about today, but if you have questions about, definitely let me know. This is called the Neisser stain. A Neisser stain helps to show granules in the Microthrix parvicella filament. So because the gram stain, it'll just look deep purple, which makes it very similar to a filament known as Nosticodia limicola 1. However, generally, Nosticodia is not a major foaming filament, so you usually don't get them mixed up in terms of symptoms on site. So generally, it's a dense foam. It's typically thought to be more dark brown, like a chocolatey color. What I will say is that the tendency of Microthrix may to be growing in more of a chocolatey brown mixed liquor. So the mixed liquor is actually what's giving the foam the color. It's not necessarily the filament. So you could have a light brown Microthrix foam or a dark brown Nocardia forms foam, but I would say Microthrix tends to prefer to grow in systems that consequently also have this dark brown color, which is why they tend to be associated. Here's another example of some Microthrix foam. This is pretty typical of what I'd see in an oxidation ditch. There's a little less agitation, so the foam is not quite as thick as it could be in some other cases, but it can kind of cover everywhere, and this can also limit oxygen diffusion into the bulk liquid. Our final type of foam is a surfactant-based foam, which tends to look really white and fluffy. So this is kind of fun because it looks kind of like snow on the surface of the basin. So you can see it's this kind of white, fluffy consistency here. And in this case, it was actually frozen. You can get frozen foam on the surface of your basin, and it can have this like crusty styrofoam consistency. It's kind of nasty, and it can start to accumulate to being really thick because it's really hard to collapse a solid foam. So definitely watch out for that in the winter. But yeah, it's got a very nice white fluffiness, and that's because it doesn't really need any solids content to cause foam. The surfactant can cause foam all by itself. So to perform microanalysis on this, you prepare a slide of mixed liquor and foam. Generally speaking, when you prepare the slides, a mixed liquor slide, you just want to get a small amount of liquid on the surface of the slide. Usually a small amount of mixed liquor on the slide will be like 20 microliters. You could just use a drop of it, but if you have too much, it can be a little bit harder to see the filaments because when you have a drop of liquid, it may look pretty flat visibly if you put a cover slip on it, but actually there's quite a bit of depth there, which can make it harder to focus on a specific layer. But anyway, you can prepare a slide with a cover slip of the mixed liquor. The foam, I recommend just putting a little bit of a drop of water on the slide and then dab a very small amount of solids from the foam with a pipette tip or something similar on top of that droplet of water. You want to barely see any solids there when you prepare the slide because that'll give you the best identification of the filaments present, and the solids content won't block things from view as much. Generally speaking, to make a really conclusive identification, you need 1,000 times magnification. Not everybody is going to have that on site. So if your microscope on site only goes up to about 400 times magnification, if you're not sure as to what a filament is, you don't have a lot of experience working with something, it's probably better to send it out or start looking into a higher magnification lens. But definitely there's nothing wrong with just commissioning an outside lab in order to do your microanalysis if you want to be sure as to what's going on. Nocardia forms tend to be visible on a lower magnification, as you'll see in this slide, because they have this nice branched tree structure. All over this, you can see all these little visible branches. Nocardia forms tend to be very distinctive for this reason. The only thing you might get mixed up with it is a filament called S. natans. They don't look that similar, but at lower magnifications, false branching from S. natans can look a lot like true branching. S. natans tends to be a lot bigger. So if you're comfortable identifying nocardiiforms, you probably won't get them mixed up. But if you're not real comfortable, that is a potential thing to confuse with it. However, there's no way you'll confuse the two once you do a gram stain. On the right here, you can see gram stain nocardiiforms. They have this nice tree branching pattern and they stain a great purple color. So they're very distinctive. All right, so next thing to keep in mind when doing microanalysis for filaments is you're going to want to compare mixed liquor suspended solids to the foam. Now, foam will tend to have a pretty consistent level of filaments, and the level of filaments you observe in the foam will be more related to how you prepared the slide than how the filament levels are actually on site, because the filaments all are concentrating at the surface. Even if you have a minor issue or a major issue, but you grab a little bit of your foam, it'll always look like there's massive levels of filaments there. But looking at the foam is the best way to conclusively say that a filament is causing your foam. On the other hand, if you look at the mixed liquor, you can't necessarily be as definitive with making an identification of the foam cause. But when you look at it, if you see kind of moderate levels of nocardiiforms or microthrix to high levels, you can be pretty confident that it is growing rapidly in the system. But if you see only a few or very low levels, there's a good chance it's actually disappearing out of the system. So this is something we include on our reports, and it's just a good way to kind of consider how filaments are growing in a system. It's a sludge age diagram. Now, everybody has sludge age calculations they do on site. You hold your sludge for longer periods of time, and then you can give your sludge age in days based on how much you're wasting. Generally, that is not always reflective of what we consider our microbial sludge age. The microbial sludge age is definitively identified by the oldest organisms present. And you can be pretty certain as to the sludge age based on that, as well as looking at flock structure a little bit, because as sludge age tends to get very high, flock tend to disintegrate more. But if you see some rotifers, but they're just starting to appear, you can be relatively sure you're at the high end of this healthy range. Then once you start to see a lot of rotifers, nematodes, that kind of thing, it tends to be in the unhealthy range. This is when you'll start to see exceptionally high levels of filaments, is on this little barrier between healthy and old sludge age, is when particularly nocardiiforms really like it. But generally, filaments tend to do better in higher sludge ages than lower sludge ages because they grow a bit more slowly. So I wanted to briefly touch on our filament origins test. My coworker Natalie did a presentation a little while back on our microanalysis program in more depth. But to touch on it today, we offer microscopic identification of filaments. We also look at flock structure and things like that and put together a report. So generally speaking, we would be able to identify any of the common wastewater filaments and give you a report in a couple days with the details as to what might be causing your foam or what could be causing other issues in the system. And we tend to pair this with customer phone calls when needed and consultation services to make sure that we really understand the customer's system. So we can make the best recommendation for that specific person, because it can make it a little less efficient if you're not doing a personalized recommendation. So when you take samples, this is something we have some issues with occasionally. That's why we're bringing it up here. You're going to want to fill a mixed liquor sample half full so there is plenty of oxygen in the bottle during transit, and then you're going to want to ship them overnight on ice. And the reason for that is so you can be comfortable your sample is as representative as of your site as possible when we receive it. Otherwise, potentially you can start to get inaccurate information as the age of the sample increases in the cooler and you don't have enough oxygen to sustain your bacterial population relatively well. So here's a little sample of part of our lab report. We tend to identify each of the organisms in captions like this. We have a very nice rotifer over here, but we're seeing a number of rotifers in the sample. There's a tardigrade over here. So some kind of fun organisms. I always like to see those. But we'll also include this, and then we tend to make a summary of what we see, and then we provide some recommendations, which are a combination of on-site changes, as well as product recommendations for what we think would be most efficient in that particular plant's case. So when you're thinking about filaments and controlling filaments in a wastewater system, you always need to keep in mind competitive balance in order to control the population of filaments. So you have a bunch of variables which might give the filaments or the good bacteria the advantage, which could be related to the food entering the system. It could be related to temperature. It could be related to sludge age. Generally speaking, when you have a change in the system, you tilt the balance between filaments and good bacteria, and you can end up with too many filaments very quickly if even just a slight change happens to favor the filaments versus the good bacteria, because they're going to be competing at all times. One of these is cold tolerance. Lots of people will notice, particularly Microthrix parvicella really likes the cold. There's a number of reasons for this. Generally speaking, filaments grow more slowly than flock-forming bacteria, so they require higher sludge ages in order to thrive. So people tend to hold a little bit more sludge in the winter, which also gives the filaments a little bit of an advantage. There's also a general factor, as good bacteria tend to reproduce more quickly than filaments. If you slow everything down, they tend to be closer to the same amount of time. So you're reducing the discrepancy between the growth rates in filaments as the temperature decreases. So generally speaking, the filaments will tend to get worse going into the winter because of these factors. So our cold temperature gives the advantage to the filaments. The next thing is a little less intuitive maybe, but one thing to consider with Nocardia forms is the surface area, in particular Nocardia forms, but also Microthrix. The surface area of filaments can give them an advantage in a system. Now, optimal surface area for a bacteria would actually be a free single-cell bacteria, but generally in a wastewater system with healthy flock, your bacteria are going to be all clustered tightly together in these little balls called flock, and the filaments will tend to be spreading out in the mixed liquor, which gives them more surface area and can give them. And this is one of the reasons that filaments tend to thrive, and they tend to start to outcompete everything as they extend more into the bulk liquid because they're just giving a big surface area advantage of them versus the other organisms. So we're going to say that gives the advantage to the filaments as well. The next thing, filaments are tolerant of low F to M conditions. Generally speaking, filaments tend to thrive in low F to M conditions because they tend to use more specialized food substrates, which are maybe harder to degrade for other bacteria present in the system. We also have some specific organisms that are filaments, which are known for low F to M conditions as well. The other thing to note, generally speaking, when you have low F to M conditions, you're going to be effectively increasing your microbial sludge age, which we've already talked about increases the prevalence of filaments. All right, the next thing, fats, oils, and grease digestion. This is a big one for foaming filaments. You have this competition between filaments and good bacteria. Well, good bacteria need to do more work in order to metabolize grease. Good bacteria have to first hydrolyze the solid grease in the system by excreting enzymes. This can break down the solid grease to give the bacteria more of an opportunity to work with them. Then they have to start performing this process called beta oxidation, which gradually converts the fatty acids present after you hydrolyze the fats into short-chain fatty acids, which they can begin to consume, whereas filamentous organisms use grease as a more structural component. It's easier for them to bring long-chain fatty acids and fats and integrate them into their structure, which gives them a big advantage over good bacteria in conditions with high levels of fats, oils, and greases. So this is the Microthrix cycle. It is fairly applicable to most filaments present in wastewater. So we can see in the early stages, you'll have filaments, and in fact, you'll have almost every variety of filament present in your system at the same time. So for example, you'll have a little small fragments of Microthrix. You wouldn't necessarily care about them at all at that point, but they're always there. Then you throw some fat into your system. What happens is the good bacteria aren't really that happy with getting the additional fat. They tend to just kind of sit there and be like, oh, well, we got to get to work producing some enzymes. What a pain. We're just going to grab the soluble BOD, which is easier for us to access. Whereas our filaments are like, oh, I love that. You guys aren't eating the fats? Well, I'm going to eat the fats instead. Then I can start to reproduce more quickly. You have other factors like temperature changes occurring at the same time, and you start to see the filamentous population grow. Now, this will start within flock, and then the filaments will begin to extend outside of flock into the bulk liquid and start to potentially break their way out of flock to float to the surface, or they will start to cause bulking in a system. So your filaments begin to form interflock bridging that can mess with settling, or you can produce a foam that when you have a high level of filaments on the surface, which is trapping air, fats, oils, and grease, and it'll start to metabolize on the surface, and it'll trap the air bubbles, it'll produce its own surfactants, and then you get a nice thick foam layer going. If you're not careful, you can recirculate these filaments back to the mixed liquor, and then start the whole cycle over again, gradually increasing levels of filaments to be worse and worse and worse until they're totally out of control and they're totally out competing your good bacteria. And you can end up with a mixed liquor that has more filaments present than good bacteria, which is a real disaster. So how do you control this? Well, we talked about what things tilt the scale in the favor of filaments, but you can also adjust this in the opposite direction to tilt the scales in favor of our flock-forming bacteria, which are our good bacteria in the system. So chlorinating, this is something that is commonly used. I tend to be a little hesitant to recommend chlorination in a lot of these cases, but what you should consider is chlorination is good at knocking out filaments because we're using their high surface area to its advantage. The filaments are not protected by this dense flock. They tend to be directly in contact with the chlorine, which can cause them to die in the system. This does come with significant risks, however, because chlorination is toxic to most bacteria present in the system, so it can lead to things like loss of nitrification. And if you control too many of the good filaments, it can actually cause your flock structures to start to be unstable and disintegrate. So you have a fairly small zone where you have an optimal dose of chlorine. If you apply not enough, you're not going to control the filament well, and if you apply too much, then you're going to be damaging a lot of other things present. So you have to be cautious. Nocardia in particular has an interesting case where they can actually fragment, and that can actually make them reproduce more quickly. A branched Nocardia tends to reproduce more slowly than a single cell or small fragment of Nocardia. So generally speaking, when people chlorinate for Nocardia, if they're not very careful, we tend to see a large growth. This is an interesting thing. If you're going to be using chlorination, I'm not going to say it's strictly bad. It may be a little safer in the case of Microthrix than Nocardia and there are some ways you can have good results from chlorination in your system. But keep that in mind. One thing to consider is when you waste sludge to control filaments, if you're putting it into your digester, a potential optimal place to chlorinate would actually be the decant from the digester, which tends to be low in solids but high in foaming filaments, so you tend to have the most direct contact with them, and it can prevent some of the filaments from repopulating in the system. Now, generally speaking, if you're wasting solids to control foaming filaments, it is safer to remove them from the site and put them on a drying bed or something like that, but I recognize that's not practical for everybody here, so keep that in mind. So how do you monitor this? The best way I would say would be to perform your gram stain and keep an eye on your filaments present in your system. Generally speaking, when filaments are damaged, their gram stain results tend to shift a little bit. So you can look and compare before and after and get a sense for how many of the filamentous cells are damaged through gram staining. You can also just look at the filament prevalence and see if it's decreasing. But generally speaking, when you're chlorinating, you have your fairly standard dose rate, but you may need to adjust for your particular system. But you're going to want to see at least 70% of the bacterial cells for filaments damaged if you have an effective chlorination application. And if you start going above that, you're more likely to damage your flock. If you go below that, you're not likely chlorinating enough to make a sizable dent in the filaments. So our favorite thing for people who are experiencing foaming is the physical removal of foam. You can do this through wasting, which is fairly typical. Like I said, it's better to put these in a drying bed if you're forced to waste in your digester. If you're playing to an anaerobic digester, Microthrix can actually survive and cause foaming for long periods of time in an anaerobic digester. However, Nocardia tend to break down fairly quickly anaerobically, so it might be less of a concern there. I mentioned chlorinating your digester decant to prevent reseeding filaments into your system. So that's the kind of thing you can do if you don't have a safe drying bed where you can apply your high filamentous solids to get rid of them from your system. But this is great because it stimulates the growth of new healthy bacteria and helps to decline levels of filaments. So anyway, mechanical foam removal with something like a vacuum truck is actually a great way to control filaments. They love to concentrate on the surface of the basin. So if you suck them off the surface, you're removing a lot of filaments from the system. Now, don't get discouraged by this necessarily. We're about to have the third poll posted, which is asking about vacuum trucks specifically. When you vacuum the foam off the surface, likely more filaments will be rising to the surface to replace the foam, so it may require several times of removing the filaments, and it may look like you're not achieving anything at first, but you are making progress, I will tell you. It just may not be visible immediately. So I have a couple comments on chlorination. We have a few people who say they have short-term success. We have people saying chlorination actually made the problem worse. That's good to know. We've kind of seen mixed results as well. I would say the tendency is for Nocardia foaming actually gets worse shortly after chlorination. And that's kind of what I expected with the vacuum trucks. A lot of people are able to use them, but you aren't necessarily able to use them easily. It can be a real pain. So sometimes other control strategies can be less costly and less labor intensive, and they may work very well. So the simplest way to control foaming is with the defoamer. All it's doing is it's messing with the surfactants present in the foam, and this causes the foam to collapse. They tend to work very quickly. They tend to knock back the foam in just an hour or two, but they tend to not last very long. Now, this makes them a little bit limited in their ability to deal with filamentous foam because the filaments are not going to be leaving the system. In fact, you're going to encourage them to grow more in many cases when you push them back into your mixed liquor using a defoamer. However, I think these are great if you use them as a temporary control method while you do something else to get something like filaments under control if they're working. Just a note, you're going to want to avoid using silicone-based defoamers if you can. I'd look into that. Our Defoam 3000 is a blend of essential oils. Generally, you apply any of these directly to the surface of the foam, but silicone defoamers can mess with flock structure a little bit. So these are never going to be getting rid of the underlying problem. But here you go. You have a little bit of an example. You might have several feet of foam present. You can apply some defoamer. It'll collapse. It'll go away fairly quickly. And this is terrific if you have occasional surfactant foaming in your system. You can collapse it and prevent foam from overflowing your basins and causing a major mess, that kind of thing, without doing too much of an extensive treatment present. But filamentous foam, it tends to be limited. Just another note, the higher the solids content in the foam, the more stable the foam tends to be. So defoamers tend to struggle a little bit with high solids foams in general, but they tend to be very good at collapsing less dense foams, and they can work on filamentous foaming in some cases, but it tends to be a little bit more limited because filaments are a solid in the foam that happens to be particularly good at stabilizing the foam structure. All right, so we have a quick question I'm going to answer here. How does the defoamer affect DO probes? And how about the film on instrumentation? So I haven't heard of defoamers affecting my DO probes. I actually am not sure about that. If anybody has any experience with that in the chat, definitely feel free to answer Aaron here. Yeah, I haven't heard of any problems related to Defoam 3000 affecting DO probes. Generally speaking, foam can affect DO levels, so your DO levels might look like they change quickly when you add a defoamer, but yeah, I'm not sure. All right, so while these solutions, chlorination and defoamers, can help to handle the symptoms and in some cases can tilt a competitive advantage in the favor of good bacteria over filament, the safer methods are to cause the filaments to lose their competitive edge by removing food substrates with the filaments like and the flock formers don't necessarily like to give an advantage to good bacteria. So the most direct way to do this is to have some sort of grease control or grease trap system. Primary clarifiers are great at removing grease. Grease is low density, it floats to the surface, so you usually can skim it off fairly successfully. This is not necessarily true with emulsified grease, but most other grease present. Think about that. One thing worth considering, if you get a lot of grease accumulation upstream in your sewer line, places like lift station, that kinds of thing, generally speaking, it's better to maintain low levels of grease through more frequent treatments for grease and cleaning rather than letting grease accumulate a lot and then cleaning it off all at once. That's going to feed a lot of emulsified grease into the system. And you'll also push a lot of larger grease chunks down the drain, which can cause major issues with the wastewater plant. Now, I recognize not all wastewater operators have any real control over say over what happens in lift station, but keep that in mind. We have a bug on a rope product, which is good at maintaining low levels of grease for long periods of time in a lift station. And our Greasezilla product will actively degrade it more in the sewer line versus a lot of conventional surfactant-based grease removers, basically soaps. But emulsified grease, when you're emulsifying a grease, it becomes, it's able to mix with water. It becomes more soluble. This isn't necessarily bad that you're doing it in smaller quantities because emulsified grease is a little easier for healthy bacteria to degrade than non-emulsified grease. They have more surface area contact with it. But it is still grease and it can still benefit filaments. So in the case of when you have grease entering your system because maybe your primary treatment's not working well enough to handle all the grease your system is dealing with, you can use something like our QuickSymL, which is a biocatalyst product, to help give our flock formers a little bit of a boost against the filaments. This accelerates fat cells and grease digestion. It does this by hydrolyzing fats into fatty acids and then long-chain fatty acids into short-chain fatty acids. and it tends to work with the existing bacterial population present. So you're trying to convert grease into things which are more usable by flock-forming bacteria. So here's a little quick animation. We can see our Quiczyme L blobs. I should quick note, this is not necessarily done to scale. Proteins are actually a lot bigger than fats, but that's very hard to look at if you have lots of biocatalysts are largely proteins. So if you have a big protein, it could practically just totally cover up all the fat in our diagram. Just kind of worth noting. But Quiczyme L there just performed hydrolysis, which broke a fatty acid side chain off of a glycerin molecule. Now this will happen a few more times. You'll break the other fatty acid side chains off of the glycerol. This is a glycerol which can be broken down through a process called glycolysis. It's a very quick process. It doesn't yield a lot of energy, but that's fine. Glycerol is not a problem. So then you have these long chain fatty acids, which you need to deal with. And generally speaking, your flock formers have a little bit of a struggle to deal with them because you need to go through a process called beta oxidation, which slowly breaks off 2 of the carbons in our fatty acid chain at a time, producing acetate from the fatty acid present. So if you have a C18 fatty acid, it'll convert to a C16 fatty acid and you'll get 1 acetate. Then it'll be a C14 fatty acid and you'll get 2 acetates. And the bacteria are only going to be really acting on the acetates, whereas your filaments are going to be dealing with this fatty acid and taking it directly into their structure. So our Quiczyme L will help to accelerate that process dramatically. So you can see you don't have to have as much of a struggle for the healthy bacteria in order to break down all of these fatty acids into the acetate structure. And once you have the acetate, your little healthy bacteria can just go around and start eating them and clean up your system. And there you go. Then your flock-forming bacteria have a much bigger competitive advantage against filaments, which can start to starve the filaments out of the system. So here's a quick study we did with a partner group. We work with a bunch at the University of Stevens Point. This is the Wisconsin Institute for Sustainable Technology. Sadly, their lab had to close down at the beginning of the COVID pandemic, but we've worked with them in a number of studies. In particular, this one, we were looking at the ability of Quiczyme L in order to reduce levels of fatty acids in a mixed liquor sample with added fat. And he added a wide variety of fat to the study. Here's vegetable oil. You can see we're getting sort of a peak separation between no biocatalyst and Quiczyme L at about 10 hours. This is also what we'll see in the next slide as well, but there's some variability depending on what type of fat you're dealing with in what amount of time is required for Quiczyme L to make the biggest difference in the system. But pretty much at all points, we're going to see improvements by Quiczyme L in fatty acid levels, indicating that our healthy flock formers are doing a better job degrading the fats present. So this next study was on bacon grease. Bacon grease tends to be a little harder to degrade than vegetable oil, but we were seeing improvements here as well, which got to be as big as like 40% improvement in terms of total fatty acid removal comparing Quiczyme L to a no biocatalyst sample. So we're seeing pretty good results here. This will hold true in colder temperatures fairly well also. So whether it's cold or warm temperatures, this product should still work well. So you can see with that fatty acid degradation taking place, we're tilting the advantage in the favor of our good bacteria. However, the other thing to consider here is when you're releasing a lot of fatty acids quickly, the flock formers would love to consume it as a carbon substrate, these short chain acetate molecules. But generally speaking, that is a high level of soluble carbon entering the system at once. And for that reason, we add in our Foam Buster product, which provides a micronutrient blend to allow the flock formers to do a better job metabolizing these short chain fatty acids. Generally, these products work well in combination. You provide the nutrients and you're breaking down the fats into fatty acids. That'll give your flock formers a big advantage versus filaments. So this slide quickly goes through dose rates, but for any application, you may want to consult with the sales rep or the lab to get a better sense for maybe how high the dose rate needs to be for your system specifically. If you have a bit worse of a grease problem, you may need a little higher dose of Foam Buster and Quiczyme L. If it's a less major grease problem, it may be a little lower than the standard maintenance dose. But keep these in mind when you're going to be running through this. I'd say for Foam Buster, we're talking approximately 2 pounds per day for 100,000 gallons of flow rate at first for your treatment, and then you do maintenance at 1 pound per day. And then generally for Quiczyme L, for 100,000 gallons per day, you're doing about 1 quart for an initial dose rate, and then you'll maintain the sample with about a pint do a little lower dose for maintenance than you will initially for that one as well. So this was kind of an interesting case where we were working with a dairy system to attempt to reduce their nocardiiforms levels. Now, one thing many of you may be familiar with, nocardiiforms really love, a quick answer, Thomas's question. Yeah, TGD is 1,000 gallons daily. So it's actually the same scale as the first diagram. Sorry for the inconsistency there. So dairy waste tends to be really something that makes nocardiiforms happy. You'll see nocardiiforms in virtually all dairy systems. However, a lot of the time it doesn't actually cause any problems, and that may have to do with their waste stream altering the characteristics of the nocardiiforms, or it may have to do with different strains of nocardia-like organisms showing up and having different characteristics when it comes to foaming. However, in this case, they ended up with excessively high levels of nocardiiforms concentrating in their foam. This happens pretty much every winter in this site, and generally speaking, the foam had been sticking around in previous years for around 4 months. So they were interested in doing a treatment. It was causing some messes and maintenance issues, and they didn't want to deal with it. So in this case, they applied Quicksime L, Quicksime P, and Foam Buster to the system. Quicksime P is a product we generally recommend for dairy systems in particular. Quicksime L and Foam Buster being kind of our standard foaming from nocardiiforms treatment. They also increased wasting a little bit at this point in time. So we were performing on-site observations where we take a few pictures. You can see here's an initial, this is kind of what their foam looks like, and we have an end photo as well to show how it changed on site. But we also did microanalysis after a couple weeks and then at the end of the treatment to see how the progression of filament went through. All right, so we had our initial microanalysis. We can see the flock were very small and fragmented looking. There were fairly moderate levels of nocardiiforms in the system. We were kind of seeing them all over the place. They weren't excessively high, but moderate levels generally means they are actively growing in the system. They all get pushed up to the foam, so you usually won't see too much higher than moderate levels. So we also looked at the foam. It had very high levels of filaments. So after 2 weeks on site, macroscopically, so if you were to take a photo of the plant, the foam would have looked identical. But we were able to look at the flock and notice that their structures were starting to become more condensed, and the levels of nocardiiforms in the mixed liquor appeared to be decreasing. So we were seeing that the treatment appeared to be working. Generally, doing microanalysis, you can see these changes more quickly than with on-site observations. But anyway, after 6 weeks of treatment, we noticed all of the foam had suddenly disappeared, but the mixed liquor did look a little better than after 2 weeks, but it ended up looking pretty similar. So most of the mixed liquor improvements we actually saw in the first 2 weeks, but it still took a total of 6 weeks for the foam to disappear from the basin. I would say that is fairly typical for a bad nocardiiform outbreak to be about 6 weeks to do the treatment. But you might have it go away a little faster or a little slower. It depends on if you're doing things like mechanical removal of foam, how much you're increasing wasting, how good your waste stream is for the filaments, and kind of how established the foothold is. So after 6 weeks here, we saw all of the filaments had dissipated. There was very little nocardiiforms remaining in the mixed liquor. So the plant appeared to recover, and they said they would have expected it to go on at least another month in this case. All right, so a few key takeaways here. Remember, filaments are always going to be present in small numbers, and they can perform beneficial roles in the system, but when their levels become high, that's when you start to see the major problems with filamentous organisms. Generally, foaming filaments rise to the surface due to their low density cell wall. They trap air, and that causes them to foam in a system. Defoamers can be very helpful for controlling bad foaming outbreak temporarily, but are not going to address underlying issues. So you need to be aware of that and be doing some sort of other treatment in order to take care of that. Make sure to keep your sludge age as low as possible. You still have to keep an eye on your nitrifying bacteria here. Make sure you're not washing them out when you're removing all this and you're wasting sludge. But just, keep sludge age low. That tends to favor flock formers versus filamentous organisms. If you know you have a lot of grease entering your system, generally make sure your pre-treatment is working if you have one. If you don't have one, it's something you may want to consider if there's any possible future upgrades for your wastewater plant, because it can make these foaming outbreaks a lot less likely if you have an effective pre-treatment system. But once the filaments in your system, the safest way to control them is to tip the competitive scales slightly so that your filaments get outcompeted by healthy flock-forming bacteria. All right, so does anybody have any questions? All right, so I saw somebody named Chris. Should I dose this year-round? If you're a plant who knows they're dealing with a lot of fat soils and greases, Maintenance is always easier than controlling an outbreak, so you definitely could benefit from applying all year round. But if you just expect to see foaming in the winter, I would suggest maybe starting in fall with your maintenance and maybe leave it out for most of the summer. You're not likely to have foaming then. Next question, how does your product compare with calcium nitrate? That's an interesting question. I would say calcium nitrate tends to not be used for the same applications. Calcium in wastewater treatment tends to improve flocculation. There's a process called multivalent or calcium bridging theory, where the more multivalent ions, such as calcium, in your system, the more dense floc will be. If you have higher levels of calcium relative to sodium, you'll be less likely to form foam because the filaments aren't going to have as easy time escaping floc. But this tends to be a little bit limited in its ability to handle Nocardia forms because Nocardia forms and Microthrix too are going to be collecting fat, and their low density can physically rip the floc apart, even if they're fairly well formed. Nitrate can also have some benefits in a system anyway. You could apply some calcium nitrate if you had a system that really had a thick foam layer, and it would actually help with the oxygen levels in the basin as well because of the nitrate there. Comment from Charlie here where he says it's harder to keep a young sludge age in the winter months. And yeah, that's kind of a fair point. One thing I should just kind of add to that is one thing you'll want to look at when you're increasing your sludge age going into the winter, you can actually look at your volatile solids and active biomass levels present in your system. So you can see, is your higher solids level actually equating to higher levels of active biomass? Because you want to have kind of optimal active biomass going into the winter. So you generally hold more sludge, but if you hold so much, your active biomass is actually going down. You're basically keeping dead cells that aren't really adding anything to your system over the winter months. So if you really struggle with keeping a younger sludge age in the winter without losing nitrification, I would say your best bet is to kind of keep an eye on your active biomass for a couple winters and see if there's any optimizations you can make that actually increase the levels of active biomass. You may be able to waste just a bit more than you think you can, or you may actually benefit from holding a little bit more sludge in some cases. So kind of keep that in mind. Another question here. I think we are seeding from a collection system. Have you ever done an exam on a pump station? Generally, we do not do much in the way of microanalysis in a pump station, though I will say in some cases you can have filaments growing in the pump station that can wash into your system, so you might have some benefit. It's also possible that something like plant or paper fibers could be mixing in with your grease and causing it to be more stable in your basin. So you could look at a sample from a pump station and see, is there anything noteworthy? But there is a decent chance you're not going to really see much under the microscope, particularly if it's just a blob of fat and some basic free bacteria kind of thing. How would you suggest to check active biomass other than mixed liquor volatile suspended solids? That is an excellent question. I really like a fairly simple test you can do is just an ATP test. Generally speaking, that is looking at levels of ATP within cells. So you lyse all the cells and measure levels of ATP versus ATP levels in the bulk liquid of a sample, which represents how many cells are actively dying at a time. But you can use ATP paired with mixed liquor volatile suspended solids measurement to get yourself an active biomass ratio. So Lumen Ultra has a nice ATP test kit. It's actually quite easy to use. We also occasionally run that test for customers if it feels relevant. So if you want to discuss that more after this presentation, definitely let us know. But it's the ATP test kit, it's not the cheapest thing. It's like $25 a test, so it's not too bad for purchasing the test kits and working on site. There's also some other equipment associated with it, but it will tell you your active biomass, which usually about 30 to 50% of your mixed liquor volatile suspended solids will be active biomass in a typical plant case. All right, what is a good ratio between MLVSS and MLSS? I would say the simplest way to respond to that would be you want as low a difference between them as possible. You want almost all of the solids to be mixed liquor volatile suspended solids. Everything that is not in that category is essentially useless gunk you have accumulating in your system. I would say you should be starting to get concerned if you're seeing about twice as much MLSS as MLVSS. Even like a third MLSS versus MLVSS starts to indicate you have quite a bit of kind of useless gunk accumulating in your system. All right, well, thank you all. If there are any other questions, let me know. Otherwise, if I didn't get around to something you were curious about today, definitely send us an email. You have my contact here, dan.s@teamaquafix.com, and I will answer any questions you shoot my way in a fairly reasonable amount of time. I'd expect an answer back in a couple of days at most. I'll thank you all for attending today, and I hope you have a nice rest of your week.